Good evening, everybody. On to the subject of Cosmids on Chapter 3, which deals with recombinant DNA technology. Now, cloning vectors called Cosmids can carry about 45 kb of cloned DNA and are maintained as plasmids in E. coli. Cosmids combine the properties of plasmids and bacteriophage lambda vectors. For example, the commonly used Cosmid PL-FR5 has two cos ends from bacteriophage lambda, flanking a SEA1 restriction endonuclease site, a multiple cloning site with 6 unique recognition sites, hind 2 PSD1, SAI1, BAMH1, SMA1, and EcoRI. or EcoRY, an origin of DNA replication, excuse me, and a TET T gene, figure 3.32. Pieces of DNA that are approximately 45 kb in length are purified by sucrose density gradient centrifugation and from a partial BAMH1 digestion of the source DNA, figure 3.32, the PL-FR5 DNA, which is 6 kb, is cleaved initially with SEA1 and then with BAMH1. The final 2 DNA samples are mixed and ligated. Some of the ligated products have a 45 kb DNA piece inserted between the 2 fragments that are derived from the digestions of the PL-FR5 DNA. These molecules are about 50 kb long and have cos sequences that are about 50 kb apart. Consequently, these DNA constructs are successfully packaged into a bacteriophage lambda heads in vitro, reconstituted PL-FR5 without insert to DNA. It's not packaged after the assembly of bacteriophage particles. The DNA is delivered by infection into E. coli F3, figure 3.32. Once inside the host cell, the cos ends, which were cleaved during the in vitro packaging, base pair and enable the linear DNA to circularize. This circular form is stable and the cloned DNA is maintained as a plasmid insert DNA construct because the vector DNA contains a complete set of plasmid functions. Moreover, the TET gene allows colonies that carry the cosmid to grow in the presence of tetracycline. Non-transformed cells are sensitive to tetracycline and die. A phosmid is a kind of cosmid vector that carries up to 40 kb of insert DNA and a cos site for in vitro bacteriophage lambda packaging. The difference between a cosmid and a phosmid is that the origin of replication of a phosmid is derived from the E. coli F factor sex plasmid, hence the name. The advantage of phosmids is that they are very stable single copy vectors, whereas cosmids are maintained at higher copy numbers, which often leads to deletions or rearrangements of parts of the insert DNA. Other E. coli bacteriophages have been used for creating vectors. For example, the genome of the P1 bacteriophage is 115 kb long. The P1 vector system can carry 80 to 100 kb of inserted DNA. The advantages of using cosmids and other vectors derived from bacteriophages are twofold. First, because the capacity of these vectors is greater than that of plasmids. Gene clusters and large genes can be cloned. Second, a larger insert in the cloning vector means that fewer clones of a genomic library have to be screened for a specific gene. This is the figure of the BAMH1 with the cloning process. And as you shows the DNA that has a BAMH1 fractionate by size, leading to a T4 DNA ligase, which helps to convert that in vitro phage packaging. When you finally go in for the infection that happens after the plasmid form with the cos sites inserted into the E. coli bacteria. Excuse me. Now to read the details here, a Cosmid cloning system. So this is exactly how infection takes place. You need to understand that. So a Cosmid cloning system, the Cosmid contains an E. coli region of replication, ORI, that allows the Cosmid to be maintained as a plasmid in E. coli. Two intact COS sites closely flanking a unique SEAS1 site, a unique BAM-H1 site near but outside one of the COS sites, and a TET-T gene. The source DNA is cut with BAM-H1 and fractionated by size to isolate molecules that are about 45 kb long. The plasmid DNA is cut with SEA1 and BAM-H1. The two DNA samples are mixed and treated with T4 DNA ligase. After ligation, some of the joined DNA molecules will have a 45 kb piece of DNA inserted into the BAM-H1 site of the plasmid. When this happens, the two COS sequences are about 50 kb apart. Excuse me. These molecules are packaged into bacteriophage lambda heads in vitro, and infected particles are formed after the addition of tail assemblies. Infected bacteriophage lambda delivers a linearized DNA molecule with COS extensions into E. coli. After entry into the host cell, the COS ends base pair and the DNA ligase of the host cell seals the links. The circular DNA molecule that is created in this way persists as a plasmid in the host cell. In this case, transformed cells can be identified because they are resistant to the antibiotic tetracycline. And now to table 3.8, which shows the estimated number of clones required for a 99% probability of representation of every DNA region in a genomic library for various organisms and cloning vectors. Now the organism, genome size BP, the clone BR322 5 kb, bacteriophage lambda 17 kb, these are all the number of clones, Cosmid 35 kb and BAC 150 kb. So we have 1, 2, 3, and 4 clones. So in E. coli, you have a clone through PBR322 5 kb, which is 4234. The bacteriophage lambda gives 1243, Cosmid gives 602, and a BAC gives 138. In yeast, you have the genome size at 123 into 10 raised to 7. Likewise, fruit fly 0.2 10 raised to 8. Rice 5.7 10 raised to 8. Humans is 3.3 10 raised to 9. And frog is 2.3 10 raised to 10. So, and the last one, humans, you have bacteriophages, you have the cloned aspects and Cosmid aspects and the back aspects. So please take a screenshot of this, make notes on this, they're very important for your exams. For the cloning vectors, the sizes in parentheses are the average sizes of the insert DNA. Now, high capacity bacterial vector systems. A vector system that carries very large inserts greater than 100 kb is helpful for the analysis of complex eukaryotic genomes. For example, these types of vector systems are indispensable for creating libraries for genome sequencing and for carrying one or more intact genes on an single insert. In contrast to a small insert library, a large insert genomics library is more likely to include all the genetic material of the organism with fewer clones to maintain. A low copy number E. coli plasmid vector that is based on the P1 bacteriophage cloning system has been devised for cloning DNA molecules that are from 100 to 300 kb in length. The DNA insert vector constructs of this system are called bacteriophage P1 derived artificial chromosomes. Similarly, the F plasmid, F factor replicon, 6 plasmid or fertility plasmid of E. coli, which is present at 1 or 2 copies per cell, have been used along with the LAG-ZT selection system of the PUC vectors to construct an extremely stable cloning vector that carries DNA inserts from 50 to 300 kb in length. The F plasmid based DNA insert vector constructs, which are used extensively, are called BACs. Now, the next topic will be genetic transformation of prokaryotes. Transferring DNA into E. coli. Transformation is the process of introducing free DNA into a bacterial cell. For E. coli, which is the main host cell for recombinant DNA research, the uptake of plasmid DNA is usually achieved by treating mid-long phase cells with ice-cold calcium chloride CaCl2 and exposing them for 2 minutes to a high temperature, 42 degrees centigrade. This treatment creates trench openings in the cell wall that enable DNA molecules to enter the cytoplasm. This method has a maximum transformation frequency of about 1 transformed cell per 1,000 cells, that is 10 raised to minus 3. The transformation efficiency is approximately 10 raised to 7 to 10 raised to 8 transformed colonies per microgram of intact plasmid DNA. Although 100% transformation frequency would be ideal, selection schemes that enable plasmid transformed cells to be readily identified over the drawback of a low transformation frequency. In some other bacteria, competence occurs naturally and in some cases can be enhanced by the use of specific growth media or growth conditions. These bacteria are usually easily transformed. Other DNA delivery systems must be used for bacteria that are either refractory to chemically induced competence or not naturally competent. And on to the topic of electroporation. The uptake of free DNA can be induced by subjecting bacteria to a high voltage electric field in the presence of DNA. This procedure is called electroporation, a term that is a contraction of the descriptive phrase electric field mediated membrane permeabilization. The experimental protocols for electroporation are different for various bacterial species. For E. coli, the cells, 50 microliters, and DNA are placed in a chamber fitted with electrodes, figure 3.33a, and a single pulse of approximately 25 microfarads, 2.5 kilovolts, and 200 ohms is administered for about 4.6 milliseconds. This treatment yields transformation efficiencies of 10 raised to 9 transformants per microgram of DNA for small plasmids, 3 kb, and 10 raised to 6 for large plasmid, which are 136 kb. Similar conditions are used to induce BAC vector DNA into E. coli. Thus, electroporation is an effective way to transform E. coli with plasmids containing inserts that are longer than 100 kb because an appropriate set of electroporation conditions can be found for nearly all bacterial species. This procedure has become standard for transforming many different types of bacteria. Very little is known about the mechanism of DNA uptake during electroporation, figure 3.33b. It has been deduced along the lines of the explanation of chemically induced transformation that transient pores are formed in the cell wall as a result of the electroshock, and that after contact with the lipid bilayer of the cell membrane, the DNA is taken into the cell. Figure 3.33 explained here. Electroporation A. Electroporation cuvette with a cell suspension between two electrodes B1, cells yellow and DNA red, and suspension in an electroporation cuvette prior to the administration of high voltage electric field HVEF pulses. 2. HVEF pulses induce openings in the cells, dashed line, that allow entry of DNA into the cell. 3. After HVEF pulsing, some cells acquire exogenous DNA and the HVEF induced openings are resealed. And cells, but the genetic features of the strains and plasmids are designed to select for their targeted recipient cells that receive the cloning vector. For example, one possible selection procedure uses a helper cell, E. coli, that maintains and now conjugation for some bacteria. The natural system of transmitting plasmids from one strain to another has been used to transport a plasmid insert DNA construct from a donor cell to a recipient cell that is not readily transformed. Some plasmids are genetically equipped to form cell-to-cell junctions through which plasmid DNA is transferred from one cell to another. And now for some bacteria, The natural system of transmitting plasmids from one strain to another has been used to transfer the plasmid insert DNA construct from a donor cell to a recipient cell that is not readily transformed. Some plasmids are generally equipped to form cell-to-cell junctions through which plasmid DNA is transferred from one cell to another. Effective contact between a donor cell and a recipient cell depends on plasmid genes that encode conjugative functions. Moreover, the mechanical transfer of the DNA requires plasmid genes that encode mobilizing functions. Most of the plasmids that are used for recombinant DNA research lack conjugative functions, and therefore they are not passed to recipient cells by conjugation. However, some non-conjugative plasmid cloning vectors can be mobilized and transferred if the conjugative functions are supplied by a second plasmid in the same cell. In other words, by introducing a plasmid with conjugative functions into a bacterial cell that carries a mobilizable plasmid cloning vector, it is possible to transfer the plasmid cloning vector to a recipient cell that is difficult to transform by other means. The typical experimental protocol for this procedure entails mixing 3 strains together. When the cells are close to each other, the conjugated plasmid, which in this case is also mobilizable, can be self-transferred to the cell with mobilizable plasmid cloning vector. Then, with the help of the conjugated plasmid, the plasmid cloning vector is transferred to a targeted recipient cell. All possible combination of plasmid transfer occurring among the components will be explained shortly after explaining this figure. Figure 3.34 is a tripartite mating. A helper cell self-transfers a conjugated mobilizing plasmid with a TET gene to a donor cell. The plasmid from the helper T cell provides conjugative functions for the non-conjugative conjugated mobilizable plasmid of the donor cell, which carries a kanamycin resistance CAN-T gene and enables transport of the latter plasmid into the recipient cell. Unlike the recipient cells, neither helper nor donor cells can grow on minimal media, and they are resistant to kanamycin and sensitive to tetracycline. The selection strategy identifies cells that are able to grow on minimal medium. In this example, the cloning vector is transferred from E. coli to P. putida. Although it is not shown, each plasmid has an origin of replication. The plasmid from the donor cell replicates in the recipient cell and to the cells, but the genetic features of these strains and plasmids are designed to select for the targeted recipient cells that receive the cloning vector. For example, one possible selection procedure uses a helper cell E. coli that maintains a conjugated mobilizable plasmid with a TET-T gene and cannot grow on minimal growth medium. And a donor cell E. coli that also cannot grow on minimal growth medium and carries the non-conjugated mobilizable plasmid cloning vector that has a kanamycin resistance gene, and a recipient cell, Pseudomonas putida, that can grow on minimal growth medium, has no incompatible plasmid and is sensitive to both tetracycline and kanamycin. After the conjugations are allowed to occur, the cells are grown briefly in complete growth medium in the absence of antibiotics before being transferred to a minimal growth medium with kanamycin. All right. And occasionally, the targeted recipient cell may receive both types of kanamycin. Occasionally, the targeted recipient cell may receive both types of plasmids. I'm sorry, that's a correction. However, this rare event can be detected by replica plating onto minimum medium with both tetracycline and kanamycin. Colonies that are formed in the presence of both antibiotics acquire 2 different plasmids, and those that grow only when kanamycin is present have the cloning vector. Since the transfer of plasmid DNA requires conjugation among 3 bacterial strains, the process has been designated tripartite mating. Now we're on to the summary of the entire chapter on recombinant DNA technology. Recombinant DNA technology comprises of a battery of experimental procedures that are used for inserting DNA segments from one organism into a vector, often a bacterial plasmid, and perpetuating the insert DNA vector DNA combination in a host cell. Remember this much for a definition or an explanation of what exactly is recombinant DNA technology. Right? Large amounts of the insert clone DNA can be retrieved when required. The process would not be possible without type II restriction endonucleases that cleave DNA molecules reproducibly into fragments of discrete sizes. These enzymes bind to specific sequences within a DNA molecule and symmetrically cut phosphodiester bonds of each strand at the recognition site. In addition, many of the enzymes such as T4 DNA ligase and DNA polymerase are important for cloning agents. A representative gene cloning experiment has a number of steps. First, DNA is isolated from an organism that contains a target gene and is cut with a restriction endonuclease. 2. A DNA cloning vector is cut with the same restriction endonuclease to digest the source DNA. The cloning vector has only one of these restriction endonuclease sites. 3. The two DNA samples are mixed with T4 DNA ligase and various combinations of DNA molecules, including vector DNA and DNA from the source organisms, are generated by the enzymatic formation of phosphodiester bonds at the ends of DNA strands. 4. Host cells, usually E. coli, are transformed with DNA molecules from the ligase reactions, which produces some cells that carry vector insert DNA constructs. Because the vector has a DNA sequence original replication that enables it to be replicated in the host cell, the entire construct is perpetuated. Uptake of DNA by E. coli is facilitated by calcium chloride heat shock treatment, electroporation, or other means. Conjugative and mobilization functions of plasmids are used in some cases to transfer a plasmid gene construct to a bacterium that is not readily transformed. Finally, selection schemes are available for identifying cells with vector insert DNA constructs and transformed cells are distinguished from non-transformed cells by testing for resistance to specific antibiotics or by observing specifically colored colonies. Cells with a specific clone target gene are identified by DNA hybridization with homologous or heterologous probe by immunological determination of an encoded recombinant protein by the presence of a specified enzyme activity or by functional genetic complementation. The probability of cloning a complete gene is increased by partially digesting the source DNA with a restriction endonuclease and forming a library of clones consisting of overlapping sequences of an entire genome. Vectors based on bacteriophage, lambda, bacteriophage B1, B1-derived plasmid B1 artificial chromosome, and F plasmid BHE have been developed for carrying large pieces of DNA and constructing genomic libraries from both prokaryotic and eukaryotic organisms. To obtain DNA segments that encode eukaryotic proteins, purified mRNA is used as a template for the enzyme reverse transcriptase to synthesize a cDNA strand. In turn, this strand acts as a template for DNA polymerase to produce a second cDNA strand. After enzymatic treatment, the double-stranded cDNA can be cloned into a vector. Multi-step strategies have been devised to increase the likelihood that only full-length cDNA molecules are synthesized and cloned. And to the last section, we have the review questions. Right, the first question will be, what are type II restriction endonucleases? Why are they important for recombinant DNA technology? 2. When circular double-stranded DNA from the plasmid P cell 1 is digested by various restriction endonucleases and combinations thereof, the following bands with lengths given in kilobase pairs are observed. You could take a screenshot of this entire question and then answer the question. Construct a restriction endonuclease enzyme sitemap with this information. 3 would be describe how plasmid PBR322 is used as a cloning vector. What are its special features? And describe the principal features of the PEUC cloning system. 5. A genomic library of a prokaryote organism is often constructed by cloning the products of a SAU3A1 partial digest of the genomic DNA into BAMH1 side of the vector. Why are 2 different enzymes used in this experiment? B. What is a partial digestion? How is it performed? C. Why is partial digestion often used for constructing genomic libraries? C. And 6. For gene cloning experiments, why is the cleaved plasmid DNA often treated with alkaline phosphatase prior to the ligation step? 7. Suggest a few different ways that recombinant plasmids can be introduced into gram-negative bacterium such as E. coli. Outline some of the strategies that are used to detect a clone target gene within a library in E. coli. What conditions can be satisfied for each type of assay? What is a cDNA library is the 9th question, and the 10th would be why would you use a plasmid, bacteriophage, lambda, cosmid, or a BAC as a cloning vector. What is the 11th would be what is a common mode of action of type 2S restriction endonuclease? What is a replica plating? Outline the basic methodology for replica plating. 13. What is a plasmid? 14. What is a tripartite mating? And 15 was what is the role of RNA-H during cDNA synthesis? So that winds up this chapter on recombinant DNA technology. For any doubts that you may have, please post your doubts and queries, and any clarifications required shall be given on the comment section. You could also link with me on my WhatsApp number 9188661251, or write to me on my email as well on newagedentalrehab@gmail.com. Alternative email will be drjacob_bk@yahoo.com. So take screenshots, prepare well. I'll be back shortly with the next chapter in the next lecture. Thank you. Bye.